Move on to our talks. The next speaker is Holger Fröhlich from University of Bern.
Hello everyone, and thanks a lot for the possibility to present our work here. So this work is essentially a master thesis. So it was mostly with work by Shashank Khanna. And this work is all part, essentially, of an EU project, Ethionomy. So this morning already several people talked about Alzheimer's disease. So one of the complicating things with Alzheimer's disease is that it is not only the case that we don't know really the true causes of the disease, but also so that this disease presumably starts decades before it actually becomes symptomatic in any case. So now obviously if people have already severe symptoms and you start treating a patient, so then this is of course relatively less effective because what has already died as brain material can never be restored with any sort of therapy. So therefore, one of the things that people are looking at and that we had tried to do here was to develop essentially a predictive algorithm that allows to predict the transition from pre-symptomatic or mild cognitively impaired patients to Alzheimer's disease. So on the other hand, the question is then always if you have such a model which does some sort of prediction, and I will show you later on what the data is that we are using here. So this is quite multimodal and also mild in the biological sense, multi-scale data. So there's of course always the question then, so can we gain a certain, let's say, at least partially mechanistic understanding of what this model does and what are essentially the interplay of driving factors for this conversion. The question is, of course, so how does this distinct from previous work that has been done? And in fact, most of the work that has been done in the field has mainly focusing on discriminating normal mild cognitively impaired and Alzheimer's disease patients. So hence more focusing also on this discriminative aspect, trying to find certain mechanisms of the disease itself. There's comparably less work on the question of conversion. So what drives the conversion of, let's say in this case, mild cognitively impaired to Alzheimer's disease patients. So here one work used and developed a certain method, so a Bayesian Cox regression model, and that was in contrast to what we did here, mainly based on neuroimaging features. So the people here did a lot of imaging processing, which we did not, and comparably only 2 SNPs in fact. So which is of course then limiting in some sense the mechanistic insight that can be gained. So one of the big issues also in Alzheimer's disease is when you now want to develop a model, you need data, and data in that field is scarce. So this is very different, like for example in the cancer field where you have TCGA and all these consortia which allow you to access large amounts of data. So here this is comparably scarce. So fortunately one of the initiatives that has been set out already in 2004 is ATNI, which is a multi-center longitudinal clinical trial with the initial goal to establish standards for obtaining and interpreting brain images and biomarkers. Enrolled into this study are normal subjects, so basically subjects which don't have any dementia symptoms, but of course they may develop them. Mild cognitively impaired people and subjects with Alzheimer's. The data is quite rich, so it contains what I would summarize in the following as clinical variables, so demographical information, different neuropsychological assessments, neuroimaging features, for subsets of patients also CSF biomarkers, and genomic data. So basically we have on one hand chip-based SNP data, and again, yet only for a subset of patients, genome sequencing data. The data has been used quite heavily in publications so far. So before I go on and present some of the results that we have been achieved with the help of these data, I should point out initially some limitations that these data has. So one being that the study represents primarily an amnestic population, so rather than an epidemiological selected population, so the population as such could be biased. Second, if you look at the age range of the people that are enclosed into the study, these are typically old or older people. And therefore, the study population as such is probably too late to find very early, let's say, hints for Alzheimer's disease or very early stages. And on the other hand, of course, like any clinical trial, you have only a limited time period until the end of the study, and therefore, for several subjects, so for example, for those which were initially normal, it is difficult to see basically any conversion to Alzheimer's disease later or to understand pathological sequences of events occurring during the disease. So the study has broadly the following design. So we have two different main phases, ETNI-1 and ETNI-2, and in between an ETNI-GO phase in which patients have been recruited. And you can see here also at which cases here different types of data have been collected, so MRI and fMRI data, so from which neuroimaging features have been processed, and PET scanning results, and also different neurological exams and so on, so which is all shown here. Oh, I have also a pointer, so which is so which is shown here, for example, in this table here. So altogether, there are around 70 neuropsychological assessments, CSF biomarkers, and neuroimaging features, and of course, demographic information. Unfortunately, SNPs have been called with different arrays in both ETNI-1 and ETNI-2/GO phases, so there's only an overlap of around 300,000 SNPs. And if you think about the let's say, clinical data, so non-genomic data, let's say, there are, of course, as always with these type of data, an issue with missing values. So now the overall approach that we took here is summarized here in this picture here. So on one hand, what we have trying to do is to say, to impose a certain amount of background information here. So we have first used, on one hand, a text mining-based tool called Skyview, and on the other hand, DisGeneNet and the GWAS catalog to collect a set of putatively disease-associated SNPs. Then second, we performed a further extraction of genomic features, so with the goal on one hand to capture aspects of the global population structure, as well as, and I will come to that later, to let's say putatively understand something about the pathway impacts on genomic level. Then we have applied some machine learning, and then basically in a later step then try to understand dependencies between most predictive factors. So we essentially used Bayesian networks for this focus, or better say an ensemble of Bayesian networks, and then extracted certain dependencies or edges which appear stably, and then try to link back or validate some of these edges and relationships that you find in Bayesian networks with biological mechanisms. So now let's go a little bit more into detail. So this slide here summarizes, still on a relatively high level, the genomic feature extraction. So we have here altogether around 300,000 SNPs in these data from both ADNI1 and ADNI2/Go phases. So as I said, so we have a set of SNPs so which came from essentially literature knowledge. And of course, there's a certain LD pruning step. So then another thing was to capture somehow aspects of the global population structure, so which we did here as often done with PCA. So the other thing was then, however, this other branch here in which we basically performed on one hand a gene mapping of SNPs, so one which we did in two ways. So on one hand we looked at LD blocks and then used dbSNP to map to genes, and on the other hand, we used EQTL and EQTL database gene tags. And based on that, we could then map to pathways and look also at the functional impact, predicted functional impact of SNPs, and then came up basically more experimentally also with a score of how basically SNP would impact the whole pathway. So this again gives an overview about the data. So we have here on one hand the clinical features, certain pathways, we have SNPs as such and certain principal components. So we reduce the dimensionality of the data essentially significantly. And we developed a model now to predict the time to first AD diagnosis. For this purpose, we used gradient boosting, simply for the reason that this is a decision tree based approach, so we could quite well here integrate data that is quite heterogeneous, has very different numerical scales, and allows us to deal with missing data automatically and allows us to also have an embedded feature selection. This approach worked quite nicely, so we got a quite good prediction performance, also compared, in fact, to, for example, random survival forest, or also, by the way, to elastic net regression. And we clearly here see also when we plot the prediction performance over time, a clear outperformance, for example, of a Kaplan-Meier plot. When looking at the top relevant features and also stably selected features, one of the interesting aspects was to find also here cell cycle as a pathway that is positively impacted. And this is interesting because it has actually been hypothesized that the aberrant re-entry of different neuron populations into the cell cycle is one of the possible disease causes. Can also stratify, of course, patients by the model, so I will go now a little bit faster due to the lack of time, but what we now did was also to, as I said, learn an ensemble of Bayesian network structures in order to see what could be dependencies between different features, and this here just shows two example views into this, or zooms into such a network structure, which highlights on one hand different now brain regions and their connections here with, for example, neuropsychological assessment scores. On the other hand, also the dependencies of those on, for example, ApoE4, which is one of the major risk factors. And ApoE4 then, as seen on the right-hand side picture, it's dependent here on cell cycle. And we partially validated some of these interdependencies by looking at the overlap with pathway interactions based on the significant overlap of contained gene sets. Then second, what we did is to map some of these pathways then to something which was completely independently developed, namely a set of cause-effect relationships that is truly based on literature mining, has been curated manually, and contains essentially interdependencies between different biological entities that are putatively causally related to Alzheimer's disease, and that has also been mapped then to certain gene sets. This is all completely independent, and what we developed is then essentially an approach which maps now between basically the Bayesian network that we have to some of these mechanisms. To sum up, what we did here was, on one hand, to take multi-level baseline data here, so of around 900 patients, to develop a predictive model for Alzheimer's disease risk, so which achieved a relatively nice prediction performance, and to investigate feature dependencies with an ensemble of Bayesian networks, and to basically then correlate some features of these Bayesian networks based to literature-derived disease mechanisms. Of course, this can only be seen as a first step, so we need to validate this independently in another cohort, so that's clear. So, of course, I'd like to thank all the people that contributed to this work, as I already mentioned, specifically Shashank, who did this master thesis around this, and thank you for your attention, and happy for any questions.
Thank you, Holger. We have time for one question because we are running a bit tight. If you have any burning question. If not, let's next move to the next speaker.